Okay, hello everyone. I think it's time to start. And before we start with our talk, there are a few announcements. So next week we have a hybrid talk by Agatha Burian. This talk will be about leaf dorsal ventrality and will be happening at Sainsbury Lab. And after the talk, or like in the evening on that day, we'll also have a dinner with the speaker. And there's an opportunity for members, for people in Cambridge to join for this dinner. And if you want to join for that one, in the last email that we sent around, if you answer to that one, then you're on our list. And basically it's in the first come, first serve base, who can join for this dinner. And this dinner is supported by the CPB, the Center for Physical Biology here in Cambridge. Now, today in the Morphogenesis seminar, we have a talk by Clare Buckley and a few more organizational things. You can ask questions during the talk, if there's something very urgent you want to know. And otherwise, of course, after the talk, we have a lot of time to discuss. And also you can write questions in the chat for after the talk. Additionally, we also have the breakout rooms as usually. So if you want to talk to Claire a bit more like private setting, then there are the breakout rooms. And now a few words about Clare. Clare Buckley, she studied biological sciences in Oxford, made her undergraduate there, and then came to Cambridge to do a PhD with Robin Franklin. And this was together with the lab of Robin Franklin, but also a startup called Danio Labs. She then went to King's College, to the lab of John Clark, to the Center for Developmental Neurobiology, to work as a postdoctoral research associate. And then since 2017, she is back here with us in Cambridge at the Department for Physiology, Development and Neuroscience, and initially as Royal Society Dorothy Hodgkin Research Fellow. And then now she is a Wellcome Trust and Royal Society Sir Henry Dale Research Fellow. And since last year, she's also assistant professor here at PDN in developmental neurobiology. In her research, she's investigating how the apical basal polarity level sculpts the development of the central nervous system. And particularly, she combines high-resolution imaging, CRISPR, optogenetics, and approaches to study the developing zebrafish neural tube. I think today, Claire, you also talk about this, particularly about buckling and breaking the neural tube. Yeah, very excited, and the stage is yours.
Great, thanks so much for inviting me to talk. I'm really excited to do it. I'll just see if I can share the screen and start the time. And thanks for the nice introduction as well. So before I start, just to mention that I'm going to show you a sort of overview of what our lab is doing and the general questions that we want to ask and answer, hopefully. And some of it was published, a lot of it's unpublished. I really like sharing data early. If anyone has any questions or wants to work on something similar and wants to discuss it with me, then please let me know. I really like working collaboratively. So even if you're watching this on a recording, just get in touch. And as Wolfram said, feel free to interrupt in the middle if it's easier, or I'm happy to answer questions at the end. Great, so I've actually slightly changed the title. Instead of specifically talking about the neural tube, although we will talk about that a lot, I'm going to talk about building and breaking epithelial tubes. And the reason for that is that the main goal of my group is to understand how mechanics and biochemical signaling work together to shape epithelial organs in development. And the kind of long-term reason for doing that is that we want to identify mechanisms by which a dysregulation of this balance might contribute to congenital diseases. So to give you a bit of background, many organs in the body, including the brain, which we've used as an example here, first of all, start off as relatively simple epithelial tubes. Can you see my mouse, Wolfram? I'm just going to use the mouse rather than point it. Okay.
Yes, I can see it.
Yeah, it's better for movies to have the mouse. Okay, so these tubes are highly polarized. They're made up of epithelial cells that have certain proteins at the apical end, which faces the inside, and the basal end, which faces the outside. And they also have junctions, apicolateral junctions, lining the center, which is a fluid-filled lumen. And so these epithelial cells are multipotent stem cells of whichever organ they're going to make. So in the brain, they would be the neural stem cells. And as well as enabling physiological functions like making a diffusion barrier or enabling polarized secretion, it's really important for these cells to have a strict apical basal polarity, and that's important throughout the whole of development. So, for example, you have to place your certain proteins at the apical end of the cells so that you can build an apical cilia that's important for signaling during organ patterning. You need to be able to regulate your cytoskeleton, for example, to enable contractility just at the apical end and not anywhere else. And that's important for aspects of morphogenesis. And even cell fate decisions are thought to be regulated by, for example, asymmetric inheritance of polarity proteins. So the sort of structure of this tissue and also the cells that make it up are very important for their function. But how do these polarized epithelial tubes arise in the first place? Well, as I mentioned, we're currently particularly interested in understanding the process of vertebrate neurulation. So how does the neural tube that then becomes the later brain initially arise? And surprisingly, there's more than one way in which the neural tube can arise, even within the same animal. So this cartoon is meant to represent the anterior part, or the top end, of the neural tube of the amniot, and that arises by a folding mechanism. So this already polarized epithelial sheet will fold into a tube, and this is called primary neurulation, and it happens through mechanisms such as actomyosin-mediated apical constriction. So here is a cross-section through a chick showing this process over time. But if you look at the posterior neural tube within the same organism, so here in the chick again, it arises by a completely different mechanism called de novo polarization and luminogenesis. So these initially unpolarized tissue polarizes in the middle of the organ primordium and makes an epithelium that then opens by hollowing. And here is the process happening in fish. Sorry, not in fish. I'm so obsessed with fish, with chick. So very little is known about the mechanisms of this secondary neurulation, or why actually amniots adopt these two different modes of neural tube formation. But at the place on the spinal cord where the primary and secondary neurulation meet, you get quite a high incidence of neural tube defects. For example, a birth defect called spina bifida, and that arises from other areas, but a lot of it is at the intermediate region between primary and secondary neurulation. So understanding the mechanisms driving this primary opening, so it's primary folding versus secondary opening mechanisms of neurulation is really important. But it's not just the secondary neural tube that arises in this way. There's actually a really big diversity of tubes that all arise by the same basic mechanisms to begin with. So here again is the secondary neural tube, in this case in a chick. Here is a zebrafish hindbrain, which is one of our models of choice. I'll explain later. If you look in a different tissue, the mesoderm, you can see the mouse kidney nephron arises by de novo polarization. Even endoderm, so gut arises this way. And the very early amniote embryo, so this is the epiblast of the embryo, also arises by de novo polarization. So these have all got different origins, different tissues, different animals, but they all have the same link between development and physiology. To be functional, they have to make a central and continuous lumen. So that means that the positioning of this apical domain is actually really important to physiological development, but we still don't really understand how this unpolarized mass of stem cells transforms itself into highly polarized epithelium and then into an open tube. And this is the process that we're trying to understand. So broadly, we're trying to ask, how do epithelial tubes polarize in the first place? How do they open? And what happens when they break? And we're particularly interested in understanding how individual stem cells respond to and affect their neighbors in order to build whole organs. So our strategy to answer these questions is first, to understand physiological development, we look in a normal organ. So we look in an animal, in this case, a zebrafish, and we use live imaging approaches. So one of the models that we use is the zebrafish hindbrain. And this provides a beautiful in vivo model for imaging the whole process of secondary-like neural tube formation. It's not exactly the same as secondary neurulation in a human, but it's quite similar in that it occurs by de novo polarization here at the middle of the organ primordium, and then a hollowing mechanism to open it. So I'll show you a movie about that just now. So this is looking down dorsally on the brain of a zebrafish, and it's labeled with this protein PAR3. And this is a scaffolding and junctional protein that labels the apical membrane initiation site. Cells polarize de novo in the middle and then open. So here you can see a 2-hour movie showing the initiation of lumen opening in the fish. And so yes, just observing cells in their physiological environment in vivo has already told us quite a lot about the process of de novo polarization and organogenesis. So just to show you some data from when I was a postdoc in John Clarke's lab at King's College London, we looked at individual neuroepithelial cells or neural stem cells inside the developing zebrafish brain. So this is alive inside a living organ, but we're just labeling individual cells so that we can see their shape better. And we found that this PAR3 protein that I mentioned marks the apical membrane initiation site. It's actually able to localize to whichever point the cells lie over the middle of the developing organ, even if that's partway along the cell length. And this happens independently of cell division. And then this protein PAR3 localizes to the cleavage furrow of dividing progenitor cells that occur very stereotypically in the middle of this organ. And this scaffolding protein then recruits apical polarity proteins and builds neuroepithelial junctions precisely in the center of this organ primordium. And so this work demonstrated that the initiation of polarization here is actually division independent. And that was quite surprising because there's compelling evidence from the literature, mostly from cell culture, that suggests the importance of the mitotic midbody in localizing apical proteins. So this work raised the question of how do these neural stem cells break symmetry in the right place if you don't require cell division for this to happen? In other words, how do epithelial tubes polarize? So we looked again in the zebrafish hindbrain, and we wanted to know sort of how do these cells coordinate their polarization at a tissue level within a dynamically growing organ? These cells are dividing and moving. How do they know exactly where to put the midline? And looking at this early stage, this is the neural keel of the zebrafish, where cells are kind of interdigitating on either side. They haven't yet made a proper midline. But we have found that there was some cadherin-rich adhesions at this zone of interdigitation. And we also found that this scaffolding protein PAR3 appears to be localized to cell vertices during polarity establishment if you look on FAS at the developing junctions. And so that raised the hypothesis that perhaps cell-cell adhesions might be responsible for directing where the apical membrane should localize during de novo polarization. That's published. I won't go into too much detail for this paper, but just to say that whilst that hypothesis was based on in vivo work, we actually tested it in vitro. And we moved to the mouse embryonic stem cell culture system in Matrigel. So the Magdalena Zernike-Gotz lab here in Cambridge published a while ago that if you culture stem cells in Matrigel, they quite closely recapitulate mammalian epiblast de novo polarization. So we collaborated with them to set up this system in our lab so that we could compare the mechanisms of polarization in an embryonic stem cell system. And that also enabled us to study the initiation of apical basal polarity alongside the formation of the first cell-cell contacts. So here you've got individual mouse embryo stem cells that divide. They localize their apical membrane initiation site right in the middle of these cell doublets. They then form junctions and then they open via hollowing. So although the architecture is very different, the same process occurs as during zebrafish neurulation. So this is the work of Xuan Liang in my lab. So he's a postdoctoral research fellow and it's recently published. So I'll just give you the highlights of it. And what he did was to culture these cells in Matrigel and see whether they made apical membrane initiation sites or not. And so here in the control, you can see PAR3 labeling the AMIS, and you can quantify its fluorescent localization in the middle of this cell-cell contacts. And however, just like in the zebrafish, when Xuan prevented these cells from dividing and then seeded individual cells that then later touched and adhered to each other, they were still able to form this apical membrane initiation site. So it's a division independent in this model as well. However, when Xuan looked in a cell line in which the main cadherin, E-cadherin, had been removed, it's a knockout line, the cells were still able to adhere to each other because they have other adhesion proteins, but they were not able to localize an apical membrane initiation site at all. So E-cadherin was found to be necessary for AMIS localization. That was a bit confusing because actually E-cadherin is present all along the cell-cell interface. So why is that important for localizing apical membrane initiation site just in the middle here? Well, Schwann did some FRAP experiments and found that this centralmost region is actually has much more stable E-cadherin than the regions to the side. So we think that E-cadherin might be more preferentially anchored to the internal cytoskeleton at this point. This is something we're now looking into a bit further. Schwann also cultured individual mouse embryo stem cells on glass, coated either with E-cadherin or with something else like fibronectin. And he was able to show, I won't go through all of this in detail, but basically these cells were able to polarize and localize the apical membrane initiation site in the middle of the cell, as long as E-cadherin was present. But if E-cadherin was not present, they couldn't do it. And it could actually localize the AMIS even without matrigel, so even without these important signaling proteins that you find in the extracellular matrix. So it shows that E-cadherin is necessary and sufficient for localizing the AMIS. And this is the quantification. So the apical membrane initiation site localizes without division. It's dependent on E-cadherin, but how about the apical proteins that it directs? Well, Schwann made he generated a stable cell line expressing PAR6 labeled with M cherry. And these are individual stem cells that have been prevented from dividing that then will move together and touch. And you'll see that when they do that, this apical protein PAR6 moves to the point of cell-cell contact. And you can actually see the individual frames underneath. So together, these results suggest that cell-cell contact directs apical protein localization at the central E-cadherin dependent apical membrane initiation site. So what's the consequence of not having an AMIS? Well, we already know that E-cadherin is necessary for early localization of this scaffolding protein PAR3 at the apical membrane initiation site. But even by 48 hours, there's no centralized PAR3 if you don't have E-cadherin. However, E-cadherin is not necessary for the cells to undergo their mesenchymal to epithelial transition and to polarize. And at later stages of development, Schwann made the unexpected discovery that cell clusters could still make lumens even without an AMIS. But they did this by a very different mechanism. You can see that here in the control, the AMIS has opened and hollowed into a lumen, whereas here without an AMIS, it's actually made this cup-shaped epithelium that then folds closed. And you get a gradual closing of the cavity. He did some live imaging to confirm that this was the case. So here is the control with the central lumen just expanding to become bigger. And here in the absence of E-cadherin, you get a cup-shaped epithelium that closes. So it seems that he's actually changed the modality of luminogenesis just by changing the E-cadherin adhesion. And so it seems that the role of E-cadherin is specifically to localize the apical membrane initiation site, and that enables the integration of individual cell apical domains to a centralized region before lumen hollowing. So this is the cover for his publication, and I think it summarizes Schwann's main findings really well. So here you have a chimeric doublet where this cell has cadherin and this one doesn't, and you can see there's no apical membrane initiation site. Here is a normal doublet where there is cadherin in both cells and you do have an AMIS. Here is a normally opening cyst, which is opened by hollowing, and here is one in the absence of cadherin that's making a cup that's closing. So we're really excited by these results because they provide an explanation for how an organized epithelial structure can arise within the center of a very dynamic organ primordium. So you've got cells moving around and dividing, but by aligning their cell-cell adhesions and their apical polarity domains, they can unify the point at which they polarize and then make sure that their lumen is in the middle. And it also uncovered this unexpected finding that removing these E-cadherin adhesions alter the modality of lumen formation. And that suggests that there's a key role for mechanics in determining whether tubes or cavities open or close. So how do epithelial tubes open rather than fold? Well, we've recently partly answered this question in a collaborative paper with John Clark's lab, again using the zebrafish neural rod. And I'll just talk you through this figure here. It's basically looking down on the developing hindbrain of the fish. And they've made a polarized midline, which is labeled with PAR3 scaffolding protein. But in this fish, this particular section of the neural rod has not been able to localize this important apical polarity protein called crumbs. So it's polarized, but actually not opened. You can see that the lumens on either side of this region inflate and make a nice open lumen, but this one stays fused shut. And so that basically shows you that you need this apical localization of crumbs at the middle in order to enable lumen opening. And the reason that that happens is because when you have crumbs present at the midline, it is initially coincident with junctional proteins such as encodierin and PAR3. But just like in the flies, it then is responsible for displacing these junctional proteins just either side of the midline. So rather than sort of being stuck at the midline, they're displaced either side. And that then enables the cells to make a surface and open by hollowing. So we've found that, that crumbs is necessary both for making the junctions and then for separating them. So again, this is published. I'm not going to go into any more details, but that's one of the mechanisms that you need to make junctions properly in the midline. But how do these junctions play an active role in lumen opening, or do they play an active role in lumen opening? Well, here we've started by looking at the role of actomyosin contractility at junctions. And as I mentioned, an important mechanism driving folding in epithelial tubes, like in primary neuronation, is by actomyosin-mediated apical constriction. So this can be by junctional myosin or maybe networks of apical medial myosin. So if you look at the surface of a mouse neural tube that's opened, it's folded closed by primary neuronation. This is an image by Gabriel Galea's lab in UCL, and you can see that this active phosphorylated myosin is present at the junctional regions, and the contractility is important for folding. But the role of apical constriction in opening secondary neuronation is not known. So this is the work of Millie Race, a student, PhD student in the lab, and Alex Fleet, who was an undergraduate student in the lab. And Millie is also a joint student with Christian Franze. And the first thing they did was to look at the open surface of the zebrafish neural tube using a transgenic line labeled for myosin light chain. And you can see that this is also present around the apicolateral junctions, just like in the mouse neural tube. So MLC is often used as a proxy for active non-muscle myosin, but actually to know really which areas of non-muscle myosin are active, you need to stain for the phosphorylated regulatory light chain. And when they did that, you get a very different localization. So here in pink, you can see the junctions labeled with zonocludins. And actually the most phosphorylated, this doubly phosphorylated myosin is only very small amounts at the junctions, and the rest is in this big blobs in the middle of the apical ends of these cells. And actually Millie has discovered that this is actually associated with primary cilia. So we're trying to work out what's going on here, but at least it shows you that you have much less active myosin at the junctions in the zebrafish neural tubes than in the mouse. So is actomyosin contractility required to open tubes rather than fold them? Well, Millie did quite a sort of blunt experiment where she bathed embryos in blebistatin, which is a specific inhibitor of myosin contractility. And she did this throughout the whole time course of polarization and lumen opening. And surprisingly, you can actually make nice junctions and they start to open a tube, but you can see that the tube opening is clearly inhibited. This was at a level of blebistatin and a duration of time that did start to cause some cell death. So she obviously has titrated this down as well. And she still does see lumen opening defects, maybe not quite as marked as this, but it's quite clear that myosin contractility is necessary for full lumen opening. So she's looked at the width of the neural tube and also the width of the cells. And she's found that both the lumens are much smaller in blebistatin treated embryos, but also the apical basal length of the cells that line the lumen is longer. So that suggests that actomyosin mediated cell shortening is one of the mechanisms behind opening the tube. But it's not the full story because it only accounts for part of the difference in lumen size. So there must be an additional mechanism driving opening. So Millie's looked at the apical lateral junctions in blebbistatin treated and control embryos. And on a population level, there's no difference in the size of these apical junctions when you inhibit myosin contractility. So along with the finding that there's less phosphorylated myosin at the junctions, this does suggest that junctional myosin contractility might not be actively involved in lumen opening. But there were region-specific changes in apical area. So if you look at the boundaries between two, they're called rhombomeres, but two sections of the brain, there's a sort of increase in distribution of apical lens sizes when you treat with blebbistatin. And also rhombomere 4 is more affected than rhombomere 5. And also the junctions themselves do look different. Perhaps they might be under less tension, they're a bit more wiggly. So we're looking into that a little bit more to try and work out what's happening at these junctions. But broadly, they don't seem to be critical for opening the tube. So where is actomyosin contractility acting to open a tube? This is a cross-section of a light sheet movie that really got yesterday, or just analysed yesterday, just showing the tube opening over time. And we're starting to collaborate with a computer modeler to try and work out where actomyosin might be acting in order to go from this initially sort of relatively straight closed tube to an open and it's actually quite hinged neural tube. So we know from our earlier publication that apical lateral junctions have to be fully formed in the middle for the lumen to open, and that's Crumbs dependent. But the junctions themselves, the contractility of these junctions themselves may not play a major role in opening the tubes, unlike in tubes that fold. We know that apical basal tissue shortening partly contributes to the opening, and that this is regulated by actomyosin contractility. But we're also wondering whether actomyosin contractility could somehow be important in regulating the ion pump function. We know from the literature that the inflation of the lumen is dependent on pumping ions and pumping liquid into the middle to generate hydrostatic pressure to inflate the lumen. So could decreasing actomyosin contractility somehow mess up this balance? And perhaps primary cysts could be where primary cilia are involved, because there's no obvious reason why primary cilia should be important in inflating a tube. They're not motile, but perhaps the regulation of hydrostatic pressure could be one mechanism by which they're involved. So that's something that Millie's actively looking into at the moment. So I'm only going to spend another sort of 5 to 7 minutes or so chatting, but I just want to highlight the more unpublished area of our research at the moment, which is looking at how epithelial tubes are broken. So most of the experiments that I've discussed so far tell us about the necessity of proteins and signals in development. But our second experimental strategy is to directly manipulate key mechanical and signaling processes within these developing stem cells so that we can test their sufficiency for normal development, but also eventually to see whether abrogating them might lead to disease. So to do this at a subcellular scale within a whole organism, we use optogenetics. And we've now made 3 transgenic lines in zebrafish, in which we can optogenetically perturb different stages of development in vivo. And we're specifically looking at neural tube development here. So for example, we have a line in which we can move around this scaffolding protein PAR3. We should mention it's not super easy to do that, but we can do it just about. But it is a full length protein. We have another line in which we can activate RhoA signaling. And I'll show you in a minute that that seems to cause contractility, which we think is via actomyosin activation. And we have another line in which we can activate PI3 kinase. And so you can perturb these different stages of development. But today I'm just going to focus on one example, which is how to control actomyosin contractility by activating RhoA. So this works by recruiting the catalytic DH domain of the guanine exchange factor for RhoA called LARG. So this is RGF12A. And we recruit it to the plasma membrane, where it then switches RhoA from its inactive GDP form to its active GTP form. So just for those of you that are not familiar with optogenetics, this is a type of optogenetic heterodimerization where you have one molecule which we call the anchor, and one molecule which you call the bait. And you can put the anchor in whichever subcellular structure you're interested in, and your bait ideally should be free floating, in this case in the cytoplasm, and tagged with a fluorophore. And then we use the phytochrome optogenetic system, which is responsive to red light. So the optimal binding light is 633 nanometers, although it's actually sensitive to quite a wide range of light. But the nice thing about this system is that you can actively unbind using near infrared light. So that means you can very rapidly switch on or off RhoA signaling. So I initially adapted the phytochrome optogenetic system for use in zebrafish when I visited Orion Weiner's lab in UCSF. You can see the original publications here, it's sort of proof of principle. And then these are the papers from his lab that show how the system works in cell culture. But since I've been here, we've had a really nice collaboration with the Cambridge Advanced Imaging Centre. And so we've helped them to optimize a light patterning system, which is made up of 2 sets of digital mirror devices. I haven't shown the actual picture of the microscope here. This is just a cartoon, but just to show you can have a binding light and an unbinding light from 2 different digital mirror devices. And that means that you can have this inverse pattern of light. Here is light to switch on RhoA, and here is light to switch off RhoA. And that means that you can pin the activation to precisely the region of tissue or subcellular membrane that you want. And it's integrated with a spinning disc microscope, and you can control the imaging separately from the light patterning. So this is the work of Helena Krellin, another PhD student in the lab, and Todd Chengzi, who's a postdoc in Cake. So this is showing you the neural rod of the zebrafish, a single Z slice. It's starting to make the midline at this stage. Here's the outside. And in gray here, you can see this anchor that's labeling the cell membranes of these neural stem cells, these polarizing neuroepithelial cells. And here in the FIRE lookup table, you can see the RhoA guanine exchange factor, or LARG, that's distributed in all of the cells. And then this box is here where we're going to switch on activation. So when you see switch on activation, you can see that the LARG is recruited very specifically to the region of interest, just at the membranes there. So what happens? Oh yes, actually, I'll tell you how this works in 3D. So this is just to show you that you can get reasonably accurate subcellular recruitment within region of interest using this system. So just to talk you through here is your bait again. So this is the GEF in the neural rod of the zebrafish. And these are some more mosaically distributed anchors. So here, only these cells will contain the anchor that can recruit the bait. And here in white are 2 different shapes of regions of interest. And I've just zoomed in on this region here to show you that you can get quite good subcellular activation. So here, just within the region of interest of these cells, but not at the other end of the cells. But that's in a single Z slice. And this is obviously not a flat tissue. It's a 3D structure quite deep inside a brain. So what happens to this 2D recruitment over a 3D plane? Well, Todd has done a diffraction limited simulation of what this illumination would look like in 3D, taking into account the point spread function of the lasers. And so these are the two original regions of interest. Looking from the side, the actual area of recruitment is kind of like an hourglass in terms of how the point spread function happens. So you can go up and down within a 25 micrometer range to see how these binding and unbinding lights sort of blur over this range. So it's very crisp at the Z plane of activation, and then they blur out to the side. And the kind of take-home message from this is that the absolute and not relative area of blurring over Z is the same no matter what size region of interest you use. And so that means that the smaller the region of interest, the more restricted the activation is in Z. And that could be an advantage if you want to target individual cells. Whereas if you want to have a whole column of activation, you can just use a bigger region of interest and you'll automatically get a bigger column of light through your tissue. So we're just working on quantifying a bit more accurately exactly what that looks like in real life, because this is obviously diffraction limited simulation. And so the scatter of light in the tissue would actually be bigger in reality. Okay, so there's about 3 slides left just to show you what happens if you do this in a fish. So this is showing you the neural rod of a zebrafish that's about the 12 somite stage. So these are cells that are undergoing the mesenchymal to epithelial transition. They haven't yet polarized at the midline. And yeah, so this is when they're cultured under deactivating light. So you can see that the guanine exchange factor, the activating LARG, is everywhere in the cytoplasm. If you switch on the lights, it gets recruited everywhere into the membranes. This is the same embryo, and it happens very rapidly. But you can then specify, if I zoom into this region here, I've put it under the fire lookup table now. And this is the region of interest where we will activate RhoA and therefore actomycin contractility. And if I just let the movie loop, I hope you can see that when we activate RhoA, there's a quite distinct contractility of the cells. So if you look, for example, at this outside area of the neural rod, you can see that it moves in as the contractility is switched on, and then it then reverses as you switch off the binding. So this again suggests the presence of an apical basal axis of actomycin contractility that we were mentioning before. So we're starting to analyze this data, and we're doing that with the help of Guillermo Serrano and Hera from Ben Stevenson's lab. And he's helping us to use particle image velocimetry, or PIV, to extract velocity and vector fields from these images. So I'll just talk you through this. I'm going to show you the graph before the movie. Basically, this box is showing you the time at which the region of interest is active. So that means when we're switching on RhoA, and either side, RhoA is actively being switched off by the other wavelength of light. And in blue, you can see the velocity of cells within the region of interest, and in red, the whole of the rest of the tissue. And what it shows you is that very quickly after activation, you get an increase in velocity of the cells inside the region of interest and actually the whole tissue. And then very soon after you switch off the activation, the cells also speed up again. So let's have a look at this. This is a velocity magnitude field within the PIV lab of MATLAB. And here is the region of interest that we're going to activate. This is the neural rod. And if I play the movie, you'll see that when it goes on, there's a rapid movement of the cells now. And then when it goes off, the cells will also move rapidly, just very quickly. I'll show you this a bit more clearly in this last slide, which is showing you both the velocity magnitude fields, but also the vector directions in these little arrows. And you can see that before activation, the directionality of the vectors within this white dotted line, this is the bit that I analysed, is relatively distributed. It's all kind of reasonably one direction, but it's relatively distributed. Whereas when you switch on RhoA activation, there's a very robust movement of cells, both downwards and to the middle of the tissue. So that's at this angle in the rose plot. And this is actually maybe the opposite of what you might initially expect. So if we're contracting this region, if this is being activated for RhoA and actomycin contractility, you might have thought that the cells around it would be kind of pulled towards it. But in fact, it's the opposite. They're being pushed away from it. Now, this is where I could do with some feedback from the physicists in the audience. I'm kind of trying to explain it in real terms, and I think it's a bit like squeezing a tube of toothpaste, that you're sort of pinching the tissue at one point, and then the cells are being displaced downwards. But the interesting thing is that this movement is then really reversible and robustly reversible. So this is a rose plot of the first 45 seconds of deactivation, and the cells move precisely back along the route that they initially took. So now they're moving back up and outwards again. So I think, in slightly dumbed down speak, that these results suggest that the cells are loosely packed enough that they can initially behave a bit like a liquid flowing away from the region of interest that we've activated. But they're connected together enough that they do retain an elastic element, because they're able to return along the same path to their original positions when the force is removed. And like I said, I'd welcome input on the physical description of these results. But what we're trying to do with them now is to use this approach to precisely manipulate the contractile forces inside the developing brain of the fish. And we can do this in a minimally invasive way and in a reversible way, with very high spatial and temporal resolution. So first, we want to use this to describe how the collective properties of these neuroepithelial cells change over the course of the mesenchymal to epithelial transition. And that might help to explain how convergence extension movements occur at the same time as neurulation in fish, for example. And then we're also using this approach to directly test the potential role of differential actomyosin contractility in both polarizing and opening the lumen. Okay, so just to summarize, how do mechanics and biochemical signaling work together to shape epithelial organs? Well, we now know that cadherin-mediated cell-cell adhesions are important in positioning the apical domain. And we also know that if you prevent this domain from forming, it actually alters the modality of the luminogenesis. We know that crumbs assembly is necessary for forming junctions and enabling them to separate to make a neural tube. And we also know that actomyosin contractility is important, but we're not quite sure how yet. But we do now have a way of testing it, because we can now optogenetically switch on actomyosin contractility deep within the zebrafish brain. And that can help us to directly test how changes in mechanics at the cellular level can affect morphogenesis at the tissue level. And in the future, we hope to test how dysregulation of this balance might cause congenital diseases like neural tube defects. So just to thank my lab, this is us on a lab retreat with the Steventon lab in the summer, which was really fun. And any questions from you, I'd really welcome. Thank you.